Hi, I'm Lori Armstrong, a biology teacher here at Shawnee Community College, and I'm going to show you an activity you can do with common ingredients and equipment you probably already have at home. If not, you can get them at your local grocery store. And we're going to extract some DNA in honor of National DNA Day, which is Saturday, April the 25th. So what you're going to need, I'm going to use some split green peas. You can also use strawberries if you have frozen broccoli or spinach. Those would work as well. And I'm going to be following this protocol from the Genetic Science Learning Center. And it's how to extract DNA from anything living. And so you're going to start off with a half cup of these split peas. You're also going to need some dish soap and some ice cold water and a blender. If you have a Ninja, something like that, that would work as well. We're going to put half a cup of split peas into our blender, along with 1/8 of a teaspoon of table salt and our 1 cup of cold water. And then we're going to blend this up for about 15 seconds. Okay, so after you've blended together this salt and pea and cold water mixture, then you want to strain it into a measuring cup or something similar. This is just going to keep all the big chunks from getting into our blended mixture. Now the purpose of blending is to split the peas apart even further and get some of the cells loose. After we've added this to something like our measuring cup that the cold water was in, then we're going to add a tablespoon of, this is Dawn dishwashing liquid. You could try other kinds of soap. You could use another brand of dish detergent. You could try shampoo, bath soap. This is going to help break apart the membranes on the cells and allow the DNA to come out. So we're just going to mix that together gently, and we're going to let that sit for anywhere from 5 to 10 minutes, and then we'll pick back up with the rest of the procedure. All right, so we're now going to take this mixture of our blenderized peas that we've strained and added our dish soap to and stirred gently. We're going to pour it into some test tubes. Now you probably will not have test tubes at home, but you can use small glasses or vases or something like that. Just don't fill them up too much because we're going to have to add some more stuff to them shortly. Once we've poured this mixture into some small containers, we're going to add just a pinch of meat tenderizer. If you don't have meat tenderizer, you can use fresh pineapple juice, or you could even use some contact solution. The reason why we're using this is it contains enzymes, and it's also going to help get the DNA out of the cells so that we can look at it. Then we're just going to gently swirl or stir to kind of mix this up a little bit. We don't want to shake it too much because that dish soap will cause it to get real foamy. Once we've done that, then we're going to pour some ice cold alcohol into these tubes. Now, the alcohol you have at home is probably going to be isopropyl alcohol. You want to stick it in the freezer. It will get very cold, but it will not freeze solid. But the ice cold ethanol has isopropyl alcohol is going to have a greater affinity or attraction for the DNA, and it's going to help to pull it up out of this green mixture in the bottom up into the clear layer where we can actually see it. Now the way you want to add this to your containers, particularly if they're tall and skinny like this, kind of hold them at an angle and then let the alcohol gently run down the side of the container. So tall containers do work better for this, tall and skinny if you have anything like that. The alcohol is also going to be less dense, and so it's going to stay on top, it's going to float. We're going to let this sit for a few minutes undisturbed, and then we'll come back in a few minutes and see if we can identify some of the DNA. All right, after you've waited for 5 to 10 minutes, after you've added the ice cold alcohol to your tubes or containers, hopefully you'll see something like this. So all of this stuff up here in this top clear alcohol layer is DNA. Now it's not pure DNA. There's going to be some cellular debris and other stuff in there. But I think you'll be impressed at how much DNA you get from doing this experiment. And again, you can use things besides the green peas. Strawberries work great. You could try other fruits and vegetables that you may have in your house. Try different kinds of soaps and see what kind of results you get. But it's just a fun, easy activity that you can do, maybe on a rainy day when you can't get outside and enjoy nature, and you can learn more about the DNA that makes each of us who we are.